Talking about bacteria, then what are bacteria? What are these cells? So here we have a definition about bacteria, and it's a very brief but accurate prescription uh, of the bacterial cell. The bacteria are unicellular, which means they are single cells, and prokaryotic organisms. And last time we learned that prokaryotic means they don't have nucleus. They don't have nuclei that have no organelles except ribosomes. We learned that in the bacterial cell there are no cellular organelles, but they do have ribosomes. They don't have membrane enclosed nucleus, so that's the major property of a bacterial cell. So the properties, the first one is that they are prokaryotic, and then they are unicellular, and of course, because they are microorganisms, they are small. So they are small unicellular cells. And bacterial cells are relatively simple structure. So here, relatively simple structure means that compared to eukaryotic cells, their structures are simple, relatively simple, but they are also well organized. They also contain structures inside the cell. So then how big are bacteria? Or how small are they? We call them microorganisms. You want to know how small they are. Well, it's hard to say because different bacteria, the sizes of different bacteria are different. But usually the bacterial cell size is around micrometer, or we can call micrometer micron. According to our living everyday life experience, it's really hard to imagine the size on micrometer because it's so small. So our ruler, the smallest scale on the ruler is a millimeter. You can imagine that if you can divide the 1 millimeter into 1,000 parts, and each part is 1 micrometer. So it's so small, and we cannot see this size. We can see the bacteria with our naked eyes, and varies. It means the bacterial size are different between different species, and even the same bacteria when they are young or when they are getting old, the size are also different. So the size of bacteria and the shape of bacteria, because there are so many species, so many types of bacteria, their shapes are different. Some are spherical, they are round like small balls. Those bacteria, we call them coccus. Coccus means round spherical structure. This is coccus. They are spherical. And usually the diameter of those spheres are 1 micrometer. So this is coccus, or for a lot, we can call them cocci. The other, they look like rod, like this rod. And this type of bacteria, call them bacillus. This is the bacillus. And for a lot, we call them bacilli. So this is a rod-shaped bacteria, and the width of the bacteria is about 0.5 to 1 micrometer, and the length is around 3 micrometer, but this varies and depends on the species of bacteria. Some bacteria can grow much longer, like 3 to 10 micrometer long. Okay, the last group, we call them spiral bacteria. So So they form the spiral shape, and the length is about 1 to 3 micrometer, and the width is 0.3 to 0.6 micrometer. So there are 3 different types of bacteria according to their shape. And here I show you different types of bacteria in these pictures: coccus, bacillus, and spiral bacteria. Yeah, another picture to show you the shapes of bacteria. And they all look different. And according to their shape, according to their morphology, we can examine them under microscope, and according to their shape we can tell the difference, and we can identify what type of bacteria they are. Another concept of bacteria is grouping. Even though we learned that bacteria are single cell or unicellular microorganisms, but in the natural environment, bacteria are not in single cell to grow. Usually they group together, so they form clusters, they form lines. So a lot of bacteria group together and live together, and this phenomenon we call it grouping of bacteria. For example, for coccus, single cell we call them coccus, but sometimes there are 2 coccus or 2 cocci to come together, live together. This we call them diplococcus. And in this word, di means 2, so there are 2 bacteria together. Or sometimes they can form a chain like this. This is a chain of cocci. If they form a chain, we call them streptococcus. Here, strept means a chain or a line, streptococcus. Or some bacteria, some species can form cubical bundle. Cubical means that they can form like a cube, like this one. 4 bacteria come together to form this tetrad, or 8 bacteria to form this cubic. And also another group we call staphylococcus. Staphy means grapes in Greek. So staphylococcus means that these bacteria cluster together to form a bunch of bacteria like a bunch of grapes. So staphylococcus. Here this is strep, means this is a typo. I'll correct it later. So this is a grouping of coccus. And also for bacillus or bacilli, they also group together in different ways. Some can live in single, some can form pairs, some can form chains. Some bacillus are so small, they are very short, even though it looks like a rod, but it's so short that it also looks like a ball or sphere. So this type of bacteria, we call them coccobacilli. Coccobacilli means that they are bacillus but look like a sphere. Okay, I'd like to show you a short video, and this video covers the bacterial shapes, and I hope this will help you to understand what we covered just now better.
Hey Internet, welcome to another Microbiology Basics. In this video, we're going to take a look at the shapes of bacteria. So hello again, welcome back. In this video, I want to take a look at the shapes of bacteria. Now, if you haven't seen, I'm currently also running for District 127 out here in Texas. You can find more information down below, and the reason why I'm running is because of, well, our current guy did something he shouldn't have been doing, and you can see the whole video down below. It's not safe for work, however. So anyways, to get to why you're here, which is what are the shapes of the bacteria? Well, there's three big shapes, and I like to think of CBS. If you know the television network CBS, you have ABC, NBC, anyway. CBS, C, cocci, B, bacilli, and S, spirili. So cocci, bacilli, and spirili. Now the cocci is berry shaped. They're spherical or ovoid in appearance, while the bacillus or the bacilli are little staffs. They're rod shaped, and the spirili are corkscrew or have a curved appearance. So let's take a look a little bit more detail in each of these. Now here's a quick little graphic that you can see, and we're going to use this again throughout this presentation. So again, CBS, cocci, bacilli, and spirili. So looking first at the cocci, it's usually round. Now it can also be oval, it doesn't have to be just round. It can vary a little bit, so it can be a little oval. When reproducing, the cells can remain attached to each other, and they can form different types of groupings. So not only, now here's, pause for a second, not only do you have to know the shapes, you have to know the clumping, basically, that they can go into. So with cocci, you can have different types of clumping, and this will be on a test, guaranteed. You have the diplococci, which is pair. You have the strepsilococci, which I'm sure you've heard of strep before. So strepsilococci, which is chain-like, and again, you can see in the graphic there. And then you have the tetrad, which is a grouping of 4, and it's divided in, or its division in 2 planes. So you can see those pictures right there. Then you have other groupings. So for example, you have Sarcina, which is a group of 8. This is divided into 3 planes. And then you have Staphylococci, which is grape-like clusters, multiple planes. Moving to the next one, the bacilli or the bacillus. This is little staffs. These are rod-shaped, and they divide in 1 plane. And of course, we have groupings of these as well. We have the bacilli, which is just uno bacilli. Then we have diplobacilli, which is pairs joined end to end. Then, of course, we have strep, again, which is a long chain. So get this into your head right now. Strep is chain-like. Strep is a chain. Okay, so streptobacilli is a long chain, while coccylobacilli, wait, you just said coccyl was a shape. Now you're saying coccylobacilli, you're confusing me. Coccylobacilli is a kind of, it's a little bit between a cocci and a little bit between a bacilli, so it's kind of that shape in between the two. Then we have spirili. Now spirili have 3 forms on their own. They have the spirillum, which is a rigid spiral. Then you have the spirochete, which is a flexible spiral. And then you have the vibrio, which is a rod shaped with a curve. And then, of course, we have some other ones just thrown in there for fun. So we have, again, our big 3. And then we have these other ones, which is the pleomorphic. This means it changes its shape. It morphs into different shapes. We have the stella, which is star-shaped. And yes, that's actually the way you pronounce it. You have to do stella. I'm just picturing people on a microbiology exam doing this. Please, if you do it, have somebody take a picture. So that's going to conclude our quick look at the shapes of bacteria. Keep in mind, once again, we have our 3 basic shapes, the CBS, cocci, bacilli, spirili. Okay, so we have to have that down. There are 3 basic shapes. Cocci, bacilli, spirili are CBS. Then we also have to know the groupings. So if they reproduce and they stick together, we can have groupings. Remember that strep is chain-like, while staph is more of a grape-like cluster, and those will be some of the more confusing ones. Di, whenever you hear di, di, you're looking at a pair. Okay, so those should get you through your exam, specifically on the shape of the bacteria. So if you like the video, please, of course, click like and subscribe.
Okay, so basically that covers the bacterial shapes. And next, we'll learn the structure of bacterial cells. Here on this slide, it shows a model structure of bacterial cell. This is one bacterial cell model, and On this model, it shows us the essential structures and also some particular or specific structures. The essential structures are the basic structure or the important structure for all bacteria. Any type of bacteria should have these essential structures. They include cell wall, that's the wall surrounding the cell, which is more rigid, and our cell membrane, which is made of phospholipid bilayers, and also cytoplasm, that's the material inside the cell, that's what we call cytoplasm, and also nuclear material. So basically that's their genetic material or DNA or RNA there. You know, they don't have membrane bound nucleus, so they just are more concentrated at some parts of the cell. We call them just nuclear material or nucleoloid. And there are also some particular structures. Particular means those structures can be present on some species of bacteria, but not on all bacteria. That includes the capsule, flagella, pili, and spores. So we'll talk about these structures one by one in detail and learn their properties and learn how they made up. And first thing, we'll learn cell wall. Cell walls, bacterial cell walls, are made up by peptide glycan. Here, peptide glycan means that in those material it contains peptide. You know, peptide is made up by amino acid molecules, and glycan means sugar. So peptide glycan are made up by both amino acid and sugar molecules. According to the cell wall thickness and structure, bacteria can be divided into two types. One is gram-positive bacteria, one is gram-negative bacteria. Gram-positive bacteria cell wall is very thick. So you can see here peptide glycan layer is very thick. And for gram-negative bacteria, the peptide glycan layer is very thin, like this. But for gram-negative bacteria, the cell wall contains both peptide glycan and also an outer membrane. So there is another layer of membrane, another membrane outside the peptide glycan. So this is a specific structure, the characteristic structure of a gram-negative cell wall. Then we'll learn how the peptide glycan layer forms. The cell wall or the peptide glycan layer is formed by backbone. The backbone is composed of the alternating sugar molecules, and also it contains a tetrapeptide side chain. Here, tetra, T-E-T-R-A, means 4. So there are 4 amino acid peptide to form a side chain. And also for gram-positive bacteria, there are pentapeptide cross bridge. And you know the word pentagon, and that means P-E-N. Penta means 5. So that's 5 amino acids, 5 glycines, for accurate, the 5 glycines to cross-link the tetrapeptide side chain. So this is the basic components of a cell wall. So then we can see how they come together, how they form the cell wall. This is a structure, schematic structure of gram-positive bacteria cell wall. So first, the backbone. So that's the saccharide backbone or sugar backbone. There's a GM, those are the sugar molecules. And on the sugar molecules, on M, there is a side chain. 1, 2, 3, 4. This 4 colorable means the 4 amino acids to form this peptide side chain. See, this is a side chain, side chain, side chain. And also, there's a pentaglycine cross bridge. This bridge links the two side chains together. See, this bridge to link the two side chains together. So basically, this is a gram-positive bacterial cell wall. And by forming the side chain and the bridge, the peptide glycan can be very thick and very contains a lot of layers. So that makes a difference under the electron microscope we just saw. So this is a gram-positive bacteria, but the cell wall of gram-negative bacteria are quite different. So this is the model structure of a gram-negative bacterial cell wall. So that's E. coli, for example. Even though they also contain the GM sugar backbone, right? You can see here the sugar backbone. They also have the 4 amino acid peptide side chain. This one, the side chain. These color balls are the side chain, but there is no cross-link bridge between the side chain. The 5 glycine molecules to link the side chain in gram-positive doesn't happen in gram-negative bacteria. Even though some side chains can be linked directly, like this, there is no bridge between the side chains. So this makes the gram-negative bacterial cell wall very thin, and there is only like several layers or a couple of layers. So these are the most important difference between gram-positive and gram-negative bacterial cell wall. On the cell wall, there are also some specific or special components for both gram-positive and gram-negative bacteria. For gram-positive bacterial cell wall, it contains a special component called teichoic acid. Teichoic acid is also a polymer. It links to the cell wall. There are 2 types of teichoic acids. One is a wall teichoic acid. This one is covalently linked to the peptide glycan, like this one. This is the wall teichoic acid. It's linked to the cell wall, right, to the peptide glycan layer. While this teichoic acid we call the membrane teichoic acid, because it's linked to the cytoplasm membrane or plasma membrane. So these are the 2 types. The teichoic acids extend through the cell wall and constitute major surface antigens of gram-positive bacteria. So they expose themselves on the outside of the cell, and teichoic acid can stimulate our immune system to respond, to generate antibodies against teichoic acid. So they are antigens, they are antigenic. So this is a special component of gram-positive cell wall. And this is the structure of teichoic acid molecules on a molecular level. Well, you don't have to remember these structures, but keep in mind that teichoic acid is on the outside of gram-positive cell, gram-positive bacterial cell, and they are antigenic. And to study the difference, the difference of cell wall of either gram-positive and gram-negative bacteria, we can use a method called gram staining, or sometimes people call it gram stain. So gram staining was established by a scientist whose last name was Gram. That's why we call it gram staining. It's a series of application of dyes, different dyes, to the cell, and after the procedure, the gram-positive bacteria can be stained with purple color, while gram-negative bacteria can be stained with red color or pink color, so that you can tell the difference under light microscope and determine whether they are gram-positive or gram-negative. The special component of gram-negative cell wall is the outer membrane. We just learned that the peptide glycan layer is very thin. It contains only a couple of layers, but on the outside of the peptide glycan there is another membrane. This membrane we call it outer membrane. Outer membrane is also a lipid bilayer membrane. On the membrane there are protein molecules which are floating on the membrane or embedded in the membrane, and this makes the outer membrane. And also there are special molecules on the outer membrane. That's what we call the LPS, lipopolysaccharide. Lipo means lipid, poly means a lot, saccharide means sugar, so lipopolysaccharide, short for LPS, on the outer membrane. And for gram-negative bacteria, LPS is very important molecules. Another name for LPS is endotoxin, because they are kind of toxic to the host. They can cause disease in host. So we'll introduce a little bit on the LPS structure. Okay, this is the structure of LPS. It contains three parts. On the very outside is the specific polysaccharide. This specific means it's species specific. One species of bacteria will have the same specific polysaccharide. Another species of bacteria will have a different specific polysaccharide. And this part we also call it the O antigen of a bacteria, O antigen. And this specific polysaccharide can be antigenic. Under the specific polysaccharide, this part is also a polysaccharide. This, because it's in the middle, we call it the core polysaccharide. The core polysaccharide has a genus specificity. So one genus will have the similar core polysaccharide, and at the root of the molecule, or on the very end, inner end of the molecule, is lipid A. Here, this is lipid A, and lipid A is associated with the toxicity. Toxicity is caused by the lipid A component, and it has no specificity. So any kind of LPS or LPS molecule from any bacteria contains lipid A, and they all can induce some pathogenic reaction in the host. So this is the basic structure of LPS. Okay, another structure we'll introduce here is the periplasmic space. Periplasmic space is the space between the outer membrane and the inner membrane. So this is the outer membrane, this is the inner membrane, and the space between them is called periplasmic space. In this periplasmic space, you can find this layer of peptidoglycan. Okay, except for this peptidoglycan, it also contains important enzymes and protein molecules and other molecules in the periplasmic space. Okay, then you want to know what are the functions of a cell wall. The cell wall does play important roles for the bacteria. For example, the cell wall can maintain the cell's shape, can maintain the cell's characteristic shape. You have some bacteria which is the bacillus and it's rod-shaped. It's only because it has a cell wall it can maintain and stay in a rod shape. If the cell wall is gone, the shape will change. Another role, another function is that the cell wall can protect the bacterial cell from osmotic pressure. You know that the osmotic pressure from outside and inside might be different. For example, if the bacteria is in water, the osmotic pressure is low. The water will come into the cell. Without a cell wall, the cell will burst and die. But the cell wall can protect the cell from osmotic pressure. Another function is that it plays an essential role in cell division. The cell wall is involved in cell division. And also, the cell wall, we learn that it contains molecules which can be antigenic. So the cell wall is the site of major antigenic determinants of the cell surface. Okay, just now we introduced the gram staining or gram stain in just like in the picture, and this video will show us how to do the gram staining step by step, and then finally can show us the difference between gram positive and gram negative bacteria. Okay, we'll watch this video. After the video, we'll have a 5 minutes break.
Okay, so the topic of this video will be the difference between gram-positive and gram-negative bacteria. So let's get started. So here's a diagram of a typical bacteria cell, and I want to note the 3 layers that most bacteria have. Their innermost layer, colored green here, is the plasma membrane, which directs what enters and exits the cell. It's a phospholipid bilayer with some proteins scattered in between the phospholipid bilayer. The middle layer in gold, this is the cell wall, which provides structural support to the cell. Made up of a polysaccharide called peptidoglycan. The cell wall is really the most important of the 3 layers when it comes to understanding gram-negative and gram-positive bacteria. The third red outer layer, this is the capsule. It's more of a sticky outer layer for attachment and protection. But like I said, I really want you to focus on the cell wall. So let's get started. So let's do a little compare and contrast, gram-positive versus gram-negative bacteria. Gram-positive, they have an innermost plasma membrane, and there it is in my drawing. And the gram-negative bacteria also have an innermost plasma membrane. So there it is. So there's our phospholipid bilayer, the plasma membrane. Now, the gram-positive bacteria have a thick cell wall made up of peptidoglycan, and so there it is, kind of 5 layers thick right there in my animation. The gram-negative bacteria also have a cell wall, it's just thinner, so there it is, only 2 layers thick in my animation. Now again, these are both the cell wall of the bacteria cells. When we look at the gram-negative bacteria, they also have an additional outer phospholipid bilayer, an additional outer membrane. And now we come to the third layer called the capsule, the sticky outer layer. So there's the capsule for the gram-positive bacteria, and gram-negative also have an outer capsule, so there's the capsule for the gram-negative bacteria as well. Well, one thing to mention is gram-positive bacteria are more easily treatable with antibiotics, simply because gram-positive lack that second outer phospholipid bilayer there. Therefore, the gram-negative bacteria are harder to treat with antibiotics, simply because it's harder for the antibiotic to gain entrance into the bacteria cell. Eventually, we're going to talk about a gram stain, and when we do, we're going to mention that gram-positive bacteria underneath the microscope appear a violet-purple color after performing what's called a gram stain. Now, the gram-negative bacteria, after performing a gram stain, appear a reddish to pinkish color underneath the microscope, and we're going to explain why this is in just a few moments. So let's talk about this process known as a gram stain. Now, the purpose of a gram stain is to help identify whatever infectious bacteria that is getting us sick. And so the reason we need to do this is because treatment will differ based upon the outcome of this gram stain. And so when we look at the steps, we're going to break this down into a variety of steps. Number one, we're going to obtain bacteria from a culture, and using an inoculation loop, we'll transfer bacteria onto the inoculation loop, and then we're going to smear the bacteria across a glass microscope slide. So the next step we're going to do is we're going to fix the bacteria to the slide so they don't get washed away when we do the upcoming steps in the gram stain. And we're going to do that by taking our glass slide, and we're actually going to pass it over an open flame a few times, and the heat will fix the bacteria to the slide. As I said, this way they don't get washed away when we start using some of the chemicals in the latter steps. So the first chemical we're going to use is a chemical called crystal violet, and like the name implies, it has a purplish violet color. So we'll add a few drops of crystal violet onto the bacteria samples, and once we do, let's zoom on in and take a closer look at the cells of the bacteria that are trapped on this glass slide. And when we do, we see that we have the gram-positive bacteria on the left and the gram-negative bacteria on the right. Now, at this stage, we don't know which bacteria in our sample are gram-positive and gram-negative. I'm just labeling them for our ease and simplicity. So right now, all we've done is we added the crystal violet. Those purple molecules were added, and they pass through the layers of the bacteria into the actual bacteria cell. And then the next step is we're going to add iodine. Iodine kind of has a brownish, goldish, bronzish color. And here come the iodine molecules. And they actually pass through the layers, and they actually bond and bind with the crystal violet molecules that have previously entered. And they kind of clump together and make the molecule bigger. So the next step is to wash the bacteria with alcohol. Alcohol is a destaining chemical, and watch what happens to the gram-positive bacteria on the left. The cell wall will shrink, and the capsule kind of gets dissolved and washed away. Now for the gram-negative on the right, the same thing with the cell wall. The cell wall kind of shrinks, but notice how the capsule and the outer layer, the outer phospholipid bilayer, that were washed away. This is because of the dehydrative nature of alcohol. And now here's a big thing that happens. Watch what happens next. Because of the alcohol wash, because the cell wall is so thin for the gram-negative bacteria, the crystal violet iodine molecules also get washed away. So the gram-negative lose their color. Underneath a microscope right now, if we were to view the bacteria samples, the gram-negative would look transparent and have no color, but the gram-positive bacteria would appear a purple-violet color because they retained the crystal violet dye. So the next step is to apply a kind of a reddish colored molecule, a dye called safranin. And so here's safranin being added, and what happens is the red pinkish safranin molecules pass through the cell wall and actually bind with the lipids of the phospholipid bilayer. And so now we can actually see the results of a gram stain underneath the microscope. Underneath the microscope, the results appear, the gram-positive appear purple in color. Now you might be asking yourself, why is that? How come they don't appear pink? Because they have safranin in them as well. The reason is simple, that the safranin, the pink color of the safranin, is just overshadowed and masked by the deepness, the darkness of the purple crystal violet. So you don't even really see the pink safranin in the gram-positive bacteria. It's there, you just don't see it. And notice how the gram-negative bacteria, because they lost their crystal violet dye and they only have the pinkish safranin, the gram-negative bacteria underneath the microscope would appear pink in color. So here are some actual pictures of a gram-positive bacteria, notice the dark purple color, and some pictures of gram-negative bacteria, notice the reddish pinkish color. So as I wrap up this video, I just wanted to put the two side by side one last time. Pause the video if you want to read through the text again. But the reason a gram stain is so important is knowing whether a bacteria is gram-positive or gram-negative, we can then begin proper treatment. As the notes say, gram-positive are more easily treatable with antibiotics. That's because they only have the one phospholipid bilayer. The gram-negative bacteria are more difficult to treat. That's because of their dual phospholipid bilayer. So there you go. Pause the video if you're still reading, and post your comments in the box below. I hope you found this video helpful. Thanks for watching.
Okay. All right. So we'll have like a 5 minutes break. Then, so just now we talked about the bacterial cell wall, and we introduced the functions of bacterial cell wall. But we should also know that sometimes, either spontaneously or induced, a bacterial cell wall can be gone. The bacterium loses its cell wall. For example, when the bacteria is treated with enzymes or that treated with antibiotics, the cell wall will be gone. But even though those forms of bacteria don't have a cell wall, they can still grow slowly, for sure, and also divide. And this form of bacteria, we call it L forms. L form of bacteria. Both gram-positive and gram-negative can form the L form bacteria. The L forms of bacteria can be produced either spontaneously or inductively. That's what we mentioned. And it's very difficult to culture them. It's very difficult to culture them because we learn the functions of cell wall because it's important. So without the cell wall, it's not easy to culture them. But at some specific condition, the modified condition, you can still culture them. They grow slowly. Some L form can revert to the normal form upon removal of the inducing stimulus. For example, if you remove the pressure of antibiotic, some L form can go back to the normal form. The L form in host may produce chronic infection, which are relatively resistant to antibiotic treatment. That's not difficult to understand because some antibiotics targets on the cell wall, targets on the molecules on the cell wall, because those L forms don't have a cell wall. So if you use those antibiotics, it doesn't work. So this is the L form of Staphylococcus, I think. Under electron microscope, the normal Staphylococcus look like this. They are round balls, spherical spheres, but without the cell wall, they form this thread-like strange structure. So this is the L form. And we know that some chemicals, some enzymes like lysozyme, targets on the sugar backbone of a cell wall, the peptide glycan, and penicillin targets on the pentaglycine bridge, and both of them can induce the formation of L-form bacteria. And we know that penicillin is the first antibiotic discovered by humans. Okay, so next important structure we'll introduce here is the cell membrane. It's one of the essential structures. The bacterial cytoplasmic membrane is composed of phospholipids, bilayer, and also proteins floating on them. The proteins can be half embedded or fully embedded in the membrane. The membranes of these prokaryotic cells are different from membrane of the eukaryotic cells by the absence of sterols. You know, on our human cells, on our human cell membrane, we have sterols like cholesterol, and these molecules are not on the bacterial cell membrane. So that's very different. Sometimes the membrane can form a very specific structure called mesosome. Mesosome is folded cytoplasmic membrane. They fold together to form tubes, to form vesicles, and especially for some gram-positive bacteria. And this mesosome can enlarge the surface of the cell membrane. You can imagine that when the membrane folds together, it can increase the total surface of the membrane, and it can increase the activity of the metabolic activity of a bacterial cell. And this mesosome also is involved in cell division. When cell divide, the mesosome plays an important role. The functions of a cell membrane include, like, they are selective permeability. They are selectively permeable, means that some molecules can diffuse through the cell membrane, while other molecules, especially those big molecules, cannot diffuse through the membrane. And the cell membrane can transport molecules through channels. The cell membrane is the site for electron transport. If you learn biochemistry, you learn the process of respiration. The respiration happens exactly on the cell membrane, and through respiration, the bacteria can generate energy. So the cell membrane is actually the energy plant of a bacterial cell. And also through the membrane, it can secrete extracellular enzymes, for example, hydrolytic exoenzymes. The cell membrane bears enzymes and carries molecules which are important in the biosynthesis of DNA and cell wall polymers. So it carries a lot of essential molecules, or essential enzymes. The cell membrane contains transport protein. That's what we mentioned just now, the channels on the membrane that allow the uptake of metabolites and release of other substances. It can take up nutrients from the environment, from the medium, and then release like toxic metabolites outside the cell. So this is a function of the cell membrane. Inside the cell membrane surround the space. That space we call cytoplasm. A cytoplasm is not an empty space. It contains a lot of materials in it. It contains abundant water. So we know that in all type of life, the most abundant material is water. And it also contains proteins and nucleic acid, such as DNA and RNA. It contains lipids and small amount of sugar molecules and salts, inorganic molecules. The cytoplasm also contains ribosomes, plasmids, and inclusions in some bacterial cells, not all, but ribosomes are in all bacteria, because this is the essential structure, essential machinery to make protein molecules. Okay, so this is the basic structure of a ribosome. A ribosome is a machine to translate mRNA into peptide sequence. It contains two subunits, one small, one large. The small subunit is 30S, while the large subunit is 50S. Here the S is called the sedimentation coefficient, and the total molecule or the total complex is 70S. In this machinery, it contains proteins and also it contains ribosomal RNA molecules. Okay, another structure we'll introduce here is plasmid. Plasmids are small circular DNA molecules. So they are small DNA molecules. They are able to self-replicate and carry some genes, and those genes can encode some properties for the bacteria, such as antibiotic resistance or bacterial virulence. But we should keep in mind that these small DNA molecules are not essential for cellular survival. Without the plasmid, the bacteria can still survive, can still replicate. But with some plasmid in it, the plasmid can contribute to some property of the cell. So this is the plasmid. Inclusions of bacteria. So that's a very special structure in bacteria, because it doesn't form in all type of bacteria. So only at some conditions, for example, when the bacteria is grown in the presence of excess nutrients. So when the growing condition is very good, and the bacteria can sense that there are large amount of nutrients, so the bacteria can try to save some for future. So they can save some material inside their cell, and that saved material we call it inclusion, which can store energy molecules. This is the inclusion. Then we know that the bacteria also contains DNA or nuclear material, but there is no membrane surrounding the nuclear material. There is no nuclei. So we call this stuff nuclear material or nucleoid, because they are not real nucleus. There can be one to several in each cell. On the left, you can see this live bacteria, and you can stain the nuclear material in red color. And you can see that they are not everywhere in the cell, but condensed at some part of the cell. But when the bacteria is dead, they can release the DNA molecule outside the cell. This is the bacterial cell body, the dead body. Well, these threads are the released DNA. You can see that the DNA molecules are much longer than the cell body itself. So which means that in a live cell, the DNA is well organized, well packed in the cell. So it must be well organized to form a tiny dot inside the cell body. Tightly packaged DNA. Okay, so basically that's what we introduced till now are the essential structures, essential material of bacterial cells. And with the rest of the time, we'll introduce the specific structures or the particular structures of bacteria, which include capsules, flagella, pili, and spores. These structures are not present in all types of bacteria, but in some type of bacteria they might have one or several of them. The first thing we'll introduce here is capsules. Capsules are some extracellular polymer surrounding the bacterial cell. So you have a bacterial cell. On the outside of the cell wall, there is another layer of polymer, and that polymer usually is sticky and smooth. So this usually is made of polysaccharide, or sometimes it's made of polypeptide. So when the polymer is condensed and to form well-defined layer, we call it a capsule. But sometimes the capsule is so thin, it only has a thin slime layer, and that layer is loose and appears to be detached from the cell. And in this situation, we call it a slime layer rather than a capsule. We can remove the slime layer easily. The capsules usually consist of polysaccharide, just as we just mentioned, but some capsule contains the polypeptide. But also keep in mind that the capsule is not essential for cell viability. You can detach the capsule, you can remove the capsule, and it doesn't kill the bacteria, and the bacteria can still survive and can still replicate without a capsule. So this is a very special structure. The capsule sometimes it's visible with a microscope, a light microscope, but to see the capsule you need to stain the bacterial cell in a special way. We won't talk about the detailed technique to stain the capsule, but keep in mind that if you stain the bacteria properly, you can see the capsule structure here. For example, this dark rod is a bacterium, and the surrounding white area is the capsule structure. The capsules play important roles for the bacteria, especially for pathogens. The first role, the first function is attachment, because it contains the polysaccharide or polypeptide. It's very sticky. It can help the cell to adhere to other bacteria or to adhere to tissue surface. It is covered with yellow color, and this yellow color is a layer of bacteria, and they stick to the teeth through the capsule. This is the first role. Second is that they protect the cell from phagocytosis. You learn immunology, you know that some cells can eat some other particles. This process we call phagocytosis. And for phagocytosis to happen, the cell needs to recognize the molecules on the bacterial surface. So without a capsule, the cell can recognize a bacterial cell and then engulf the bacterial particle. But with a capsule, the cell cannot recognize the bacterial cell anymore, so it can protect the cell, the bacteria, from phagocytosis. So first thing, this is a phagocytes. This is a bacterium without a capsule. Antibodies can recognize the bacterium, bind to the bacterium, and then the receptor on the phagocyte's surface, the Fc receptor, will recognize the Fc fragment of the antibody, and then phagocytosis can happen. This phagocytosis to take the bacteria inside the cell to form a phagosome. Then the phagosome will fuse with the lysosome. When they fuse together, the bacteria will be killed. So this is a process of normal phagocytosis. But if there is a capsule surrounding the bacterium, see here, the molecules on the surface is hidden, so your phagocyte cannot recognize the bacterial molecules. Then the bacterium is protected. So this is the second function. Another function of a capsule is that they act as a barrier. So it's thick sometimes, and it can be a barrier to antimicrobial substances. For example, the bleach or other toxic molecules, chemicals, or some substances in the blood or body fluid, such as antibiotics, complements, or lysozymes, which is the barrier for these molecules to protect the bacterial cell. Okay, another structure we'll introduce here is flagella. Flagella is the motion organ of a bacterium. Bacterial flagella are a thread-like structure. They are the organ of locomotion and provide motility for bacteria. The flagella attach to the cell surface, to the cell membrane, to the cell wall, and it can rotate at the base. And by rotating the flagella, it can drive the bacterial body to move. And this is how they call it the organ of locomotion. Flagella is composed of proteins, and those proteins we call them flagellin. And these flagellin proteins are highly antigenic. They are antigens. So we call flagella H antigens. So till now we learned two antigens on the bacterial surface. One is the O antigen, which is a specific polysaccharide of LPS, and the other is H antigen, which is carried on flagella. Flagella can be much longer than the bacteria itself. We learned the size of bacteria, which is on the micrometer scale, but the flagella can be 10 or 20 times longer than the cell itself. It can be 10 to 20 micrometer long. So it's very long compared to the cell itself. Here it shows us the flagella structure on different types of bacteria. Keep in mind that different flagella can be different according to their species. Some species has only one flagella, while others has two or even more. So this is a basic structure, schematic structure of a flagella. It attached to the surface, to the cell wall, anchors to the cell wall. It can rotate at the base, and by rotating the flagella, it can drive the cell to move. It's the organ of locomotion. They can provide motility for bacteria, allowing the cell to swim toward nutrients and also swim away from poisons. So this is it's like the leg of a human. Okay, another specific structure is pili. Pili are different from flagella because flagella is very long and kind of flexible, but pili are rigid surface structure. It attached to the surface. They are shorter, much shorter and thinner than flagella. Pili are composed of structural proteins subunits, which are called pilins. Those pilin molecules, pilin proteins, come together to form this short fiber, which are called pili. Actually, there are a lot of types, a lot of kinds of pili. Ordinary pili, sex pili, and also type 4 pili. A lot of types of pili. But we'll introduce two pili. One is the ordinary pili. Ordinary pili are short structure, and they play a role in the adherence of the bacteria to host. And ordinary pili are related to their virulence of pathogenic bacteria. So this picture shows us a bacteria with short ordinary pili on their surface that helps the bacteria to attach to the surface. Okay. And sex pili is a very special form of pili. They are responsible for the attachment of a donor cell to a recipient cell. For example, in this picture, this is a donor cell which carries ordinary pili here, the short ones, and also a sex pili. The sex pili can attach a recipient cell, and then it can pull the two cells together. When the two cells are close to each other, there will be a bridge or a channel formed in between the two cells. At that time, this donor cell can copy its DNA, either it's a chromosome DNA or a plasmid DNA, into this recipient cell. This is a very important way to transfer DNA from one cell to another, and this process we call conjugation. Sex pili are related to the transfer of drug resistance or virulence genes between bacteria. This is a sex pili structure. Okay, the last special structure we'll introduce here is called endospore. It's a number of gram-positive bacteria can form a special dormant structure under conditions of nutritional depletion or drought condition, and this is dormant structure we call endospore. One cell can form only one internal spore. When the spore returns to a favorable condition, the spore can germinate to produce a single vegetative cell, and this single vegetative cell can replicate again like normal cells. So spores are commonly found in the genera of Bacillus and Clostridium, and those we'll learn in detail in future. Bacillus and Clostridium. This is the structure of endospore formed in the cell. You can see that the endospore is much smaller than the vegetative bacteria itself, and it contains all its genetic material in the middle, and also it contains a lot of layers. Those layers can protect endospore and keep it safe for a long time. When the spores form, it can form a different position of the cell. Some spores form at the center of the cell, we call the central endospore. Some form at the terminal. This we call the terminal spores, while others can form at other places like the subterminal, close to the terminal. This one is a swollen endospore. So one end of the bacteria is swollen, bigger, different shapes, different position of the endospore. Endospores are highly resistant to environmental factors such as desiccation, means drought or dry, heat or chemical agents. So they are very stable and resistant to these agents. Endospores can exist for a very long time without dying, so they dormant and they keep quiet for a long time. So when the condition is back to normal, they can germinate to form replicating cells. Endospores usually they don't elicit disease. They only cause disease when they enter the host and germinate to produce vegetative cells. It's not easy. It's very difficult to decontaminate the endospores. Usually it requires a high temperature like 120 degree or 130 degree, and to keep the endospore at that high temperature for a long time, like 15 or 20 minutes, to kill them. So they are very resistant to these factors. I have another video to show us bacterial structure and function. Do you want to watch the video now, or you can see the video? It might take some time.
Bacteria are single-celled prokaryotic microorganisms. They can be found everywhere on the planet. The ancestors of modern bacteria appeared on Earth about 3.5 billion years ago. The cytoplasm is the basis of a bacterium cell. It contains the genetic material, that is, the ring-shaped DNA. Unlike in eukaryotes, proteins are not attached to the DNA in prokaryotes. The cytoplasm is enclosed in the cell membrane. All bacteria have a cell wall. It is tough and rigid, and it is made from proteins and polysaccharides. It provides the cells with structural support and protection. Outside the cell wall is the capsule, which is made mainly from polysaccharides. It protects the cell and is responsible for the diseases caused by some bacteria in the host organism, flagella. Bacteria have an exceptional ability to adapt and reproduce, and this is why they are extremely widespread. They are capable of both sexual and asexual reproduction. If they reproduce through binary fission, which is a form of asexual reproduction, it results in a large number of individual bacteria cells. Bacteria occur in a wide range of shapes, including spheres, rods, and spirals. Spherical bacteria, which are called cocci, can occur as single cells, pairs, chains, or clusters. Many bacteria that produce suppurative infections or pneumonia are spherical. Gonorrhea is also caused by cocci. One group of rod-shaped bacteria are called bacilli. Many bacilli cause diseases like the pathogens of plague, tuberculosis, salmonellosis, and tetanus. Escherichia coli, which is commonly found in the human intestinal system, is also a bacillus. Syphilis, Lyme disease, and leptospirosis are caused by spiral-shaped bacteria. Gram staining is a method of differentiating bacteria into two groups, i.e., gram-negative and gram-positive. The basis of differentiation is that staining produces different results in the cell walls of different types of bacteria because of their different structure. The two types of bacteria react to antibiotics differently, so treating different bacterial infections requires different antibiotics.
Okay, so that's all for our lecture today.